The MACSYS project is a research project funded by EU 7 Framework Programme under the research stream Research for the Benefit of the SMEs, Small and Medium-Sized Enterprises. It's a collaborative project between SMEs, research institutions in Denmark, Germany, UK, the Netherlands, Spain and France. In the last decades, continuous focus has been on improvement of yield and sustainability of meat production. Deboning of meat can be a technology which can minimize meat waste. Today, the equipment used to meat bone separation in poultry makes it possible to produce raw meat material which cannot be distinguished from regular minced meat. However, at the moment there are no objective methods available which measure quality of separated raw meat material which are available on the market. The aim of MACSYS is therefore to develop an efficient and objective method to classify comminuted poultry meat directly based on quality. Two main outcomes are expected from MACSYS: an automated image analysis system which can be used by food laboratories and authorities in EU to determine the level of muscle structure degradation, and a fast-track system which can be integrated on the production line and measure different quality traits of comminuted poultry meat.
We are looking at the histology to determine the level of intact muscle structure in each sample produced from the different machines and conditions. This is because the legislation on mechanically separated meat refers to the loss or modification of the muscle fibre structure in the definition. If there is loss or modification of the muscle fibre structure, then the sample is to be declared as mechanically separated meat, or MSM. The method to look at the muscle fibre structure is fairly simple but not quantitative. In the MACSYS project, we are exploring different ways to quantify the samples, and one way is to subjectively assess the muscle fibre structure under a microscope and to provide images for a software-based measurement of the muscle fibre.
At Aarhus University, they develop new antibodies for detection and quantification of muscle tissue.
Here you see a staining with our new antibody. You can notice that there's a uniform distribution of color within the whole slide, or at least what's myosin. In the previous experiments, we had to rely on two different myosin isoforms and two different antibodies. This we've overcome with a newly developed antibody. The image analysis system that we are developing in collaboration with SoftGrid is supposed to select from this large area up here, which represents the sample, the whole sample, to select individual areas within this sample and then sum up the result of the end. And this is one of the results down here of a selected area. And this is actually area 6 that we can recognize up here.
SoftGrid expects to develop an application to be used by food laboratories and legislators in order to obtain objective measures of the amount of muscle tissue and the level of degradation. The food industry could also use the program to calibrate the online sensors and machines in order to secure the final quality of the product. The business model of the final application will be cloud-based, pay per use, so that they only have to pay for its real use.
At University of Copenhagen, we explore how to determine the quality of MSM rapidly. In particular, we're testing multispectral methods as they can be implemented directly on a product stream. We use these multispectral data, like you can see an example of here, and chemometrics to develop some models that can predict certain biochemical traits that we're interested in, and that could, for example, be fat and protein content.
The fat content of the sample is measured by taking a quantity of sample, hydrolyzing it with acid to release the fat from the matrix, then drying it down and extracting the fat using solvents. This is an automated Soxhlet extractor which we use to extract the fat. Calcium and iron content of the samples is measured by ICP-OES. The samples are digested in acid and they're made up to a specific volume, and then the quantities of calcium or iron or any other mineral is measured using atomic emission spectroscopy.
The knowledge and methods obtained by the RTDs is then transferred to Cowmetec.
Cowmetec's goal is to turn an advanced and fragile lab equipment like this into a fast measurement equipment suitable for a production line.
This will be for the benefit of the poultry industry, which can optimize the quality of the products and thereby the end value of the final product. In addition, also the equipment manufacturers will be able to integrate the fast-track system on their existing equipment and thereby increase the value of their equipment.
Robert Damkjær is an end user of comminuted poultry meat and was one of the main actors in the planning of the MACSYS project together with Aarhus University. Their expectations are that MACSYS can provide tools for clear, objective quality assessments of mechanical separated poultry meat. 9 partners participate in MACSYS, 5 companies and 4 research institutions spanning the entire chain from equipment manufacturers, raw meat producers and users of comminuted poultry meat. The industrial partners in MACSYS are Robert Damkjær, Mette, Lima, Softcrit, Marel, and the research institutions are Max Rübner Institute, Letterhead Food International Limited, University of Copenhagen and Aarhus University.